Hello and welcome to this Chemistry World webinar, AI Meets Chemistry, Standigm's breakthrough with Synthia Retrosynthesis Software. I'm Natalie Starkey, Chemistry World's science media producer, and I'm here with you for the next hour. Now, please feel free to turn on closed captions if you find that helpful today. You should find the button in the bottom right of your screen. And I'll just also point out that this webinar is being recorded, so if you miss anything today or just want to watch again, then please look out in your inboxes for a link to the recording in the next few days. Now, today we're working with Merck and Standigm. Merck are actually sponsoring today's webinar, and we'd like to thank them for choosing to work with Chemistry World again. Merck are a leading science and technology company operating across life science, healthcare and electronics. From providing products and services that accelerate drug development and manufacturing, as well as discovering unique ways to treat the most challenging diseases, to enabling the intelligence of devices. And Standigm are a pioneer in AI-driven drug discovery, using cutting-edge artificial intelligence to streamline the development of pharmaceuticals. With a robust AI platform that includes biology and chemistry components, Standigm accelerates the discovery of novel drug targets and molecular designs, aiming to revolutionise patient care globally. Now, as with all Chemistry World webinars, this session is interactive, which means you can ask questions to our speakers about any aspect of what's covered today. You'll find the questions tab probably in the top right corner of your screen, but it might be different depending on which platform you've joined us on today. But please use that questions tab to send in your questions as soon as they come to mind at any point during the webinar, and I'll be able to put them to our speakers in the live Q&A towards the end of the webinar. You won't usually be able to see other people's questions in the questions box, but don't worry because myself and the Chemistry World webinars team can, so we'll be getting them all through. Now, we have two speakers joining us today, Eva Gajuska from Merck and Yu Young Song from Standigm, and they are here live to answer your questions when they come in, but their presentations have been pre-recorded. So what I'll do now is just welcome both our speakers on screen, ask them to turn their cameras on, and I'll introduce them properly once we can see them. There we go. Morning, Eva. And we've got Yu Young. Hi, good. So glad you've been able to join us today. Thank you so much for being here. Let me explain to the audience a bit about who you both are. So Eva is Head of Product Management in Merck, where she leads the development of Synthia Retrosynthesis Software. She graduated in biotechnology and chemistry from Gdańsk University of Technology and received her PhD from the Polish Academy of Sciences. Her doctoral work was devoted to development of the Synthia Retrosynthesis software for computer-assisted synthetic planning. So you can see she's going to be a very useful person to hear from today if you're interested in this topic. We're then going to hear from Yu Young, who is a principal AI scientist at Standigm, specialising in generative and synthesis AI for drug discovery. Song holds a degree in computer science and technology and mathematics and a PhD from the Graduate Institute of Ferrous and Energy Materials Technology at POSTECH in South Korea. Her work on computer-based simulations to develop novel enhanced materials has led her to dedicate herself to AI-based drug discovery. So again, we're going to hear lots from these speakers in just a minute. And if you've got questions, as I've said, please send them in because they're going to be really excited to answer those towards the end of the webinar. So please, do start sending in those questions. But first, we're actually going to have a quick poll for the audience. We've got a couple of polls today, which we'll be bringing up on screen. And what you can do is actually interact with these polls. It would be lovely to hear from our audience today. Let me launch the first poll. Which should appear on your screen now. You may need to scroll using the little scroll bar so that you can see all of the answer options. I'm not sure how it looks on your screen. It does depend what you've joined us on today. But what we're looking for is what best describes your current role. You've got lots of different options there. I can see lots of people are already interacting with the poll, so thank you very much for that. It's just nice that our speakers can kind of see who we've got in the audience today, who they're going to be addressing later on in the Q&A, and kind of who's here today. So I can see we've got actually quite a mix of people, a lot of drug discovery chemists, a lot of academics in the audience, but yeah, it's a really good mix of people, which I'm sure is what was expected for content that we're looking at today. That's brilliant. I'll close that poll. Right, well, thank you, Eva and Yu Young. We'll see you shortly. We're going to do your pre-record presentations now. We're going to hear from Eva first and then you, Young, and we'll be back in a bit. So I'm going to let my team launch the video. Thank you.
Thank you so much for this kind introduction and welcome everyone that has joined us today. This webinar will have two parts and I will be first talking about what actually Synthia is and how it can help you to overcome the synthetic challenges. I will describe a few solutions. First one is Synthia Web App, and Synthia Web App can help you with planning the synthesis both for single targets but also for the libraries of target molecules. And there are two features in Synthia I will especially focus on today. First one is called Shared Path Library, which enables to plan synthesis for the entire library of molecules using a shared synthetic route. The second one is called Diversity Library, and this can help you in the target molecule structures generation. I will also shortly describe our Synthia APIs that enable quick filtering of the molecules using Synthia Synthetic Accessibility Score, and the full retrosynthesis API that allows for connecting Synthia with your internal molecule discovery solution. With that, I will pass to Dr. Yu Yang from Standigm that will talk about how Standigm uses Synthia in their drug discovery, and will show as well some of the use case and the results. So with that, let's start. As chemists, we spend a lot of time learning all the chemical reactions, but we are somewhat biased on the reactions we use frequently, and we may not remember every possible reaction that's ever been done. And this is why we may overlook a transformation that could be really useful for a particular synthesis. Another problem we experience if we work on a target that has been already published is that sometimes the literature route is not reproducible. If, on the other hand, the target is a new molecule there would not be an exact synthesis, or sometimes even a similar synthesis at in the literature. And another thing is that the failure of the synthesis can actually happen at any step. So it would be beneficial to have something that predicts various potential pathways. Finally, it's impossible to remember the entire catalogs of molecules that you can buy. So it would be great to have something that unites all of this together. And this is actually why Synthia was designed. There are few key components of Synthia that I would like to talk about today, and these components are the base for all our pathway planning features and the molecule library design features. The first component I would like to talk about are Synthia's reaction rules. Based on these reaction rules, the software plans the synthetic pathways. So what you can see here on my screen right now is an example of one of such a reaction rule. These rules are in Synthia coded by the expert PhD-level organic synthetic chemists in a way so that they are able to consider all of the things that human chemists would consider when looking at a particular reaction. So for example, are there any functional groups that might not be compatible or that might need to be protected? Or for example, what are the structural features of the substrates that are influencing stereochemistry or regiochemistry of the reaction products? But what is extremely important here is that these rules are also written in a general way that allows them to be applied on the known molecules, but also on the completely novel targets never synthesized before. And let's now move to the second Synthia pillar, which are the algorithms that are responsible for chemical logic to design the pathways that are selective, not having any strain-related problems, or for example, highly reactive functional groups, or for example, structural motifs that under certain reaction conditions would undergo rearrangement. So actually all of these things that assure the chemical correctness of the predicted molecules and reactions. Synthia also includes the algorithms that are responsible for strategizing over multiple steps during the pathway design. So, for example, Synthia combines the steps that can be performed under the same reaction conditions, like in the simultaneous reactions, or reactions that once applied in a retrosynthetic direction can provide the possibility of applying highly simplifying transformations. And what is important here, all of these algorithms in Synthia are working together, analyzing the pathways globally, which means analyzing millions of such combinations of reactions that are possible to come up with the most optimal solution. Okay, so let's now move to the third Synthia pillar, which is the advanced path search algorithm itself. This algorithm is scoring and exploring in a retrosynthetic direction all of these possible cuts and combining these cuts into the pathways that starts from commercially available starting materials, also taking into account the user requirements. But what are those requirements? Of course, those requirements are different for different synthetic projects. And this is actually why Synthia has many customization options that one can use to adjust the results according to the project needs. And this is especially important in the context of sustainable chemistry to, for example, avoid gaseous reagents or transition metal catalysis. The user can also avoid the toxic substances or, for example, promote reactions catalyzed by enzymes. Two very recent additions in the software is that the software can consider spent-based emission factor information used for calculating the carbon footprint of the pathway. And another new thing is that the user can customize in Synthia the starting material requirements to construct the pathways starting from molecules available in custom quantities, for example, 500 grams. And what it does is, this is especially important if you're planning a multi-gram or multi-kilogram synthesis. So let's now move to how actually Synthia presents the results. And there are three ways of how Synthia presents the results. First way is the building blocks view. The second one is the bond disconnection view. And the third one is the pathway view. So this is my Synthia account. You can see here all of the analysis I performed before, and each of the styles have the small indication that it's either like a library search or a single molecule search. There's a target structure visible, and if I click on that, I will get the results for the specific molecule. This is a sitagliptin molecule, and what you can see here, this is this first view, building blocks view. You see this colorful building blocks that also are showing where this building blocks actually was incorporated into the target structure. This is a very interactive view, so we can click on the molecule to get some more details like PubChem links, like vendor information, like illustrative references. And by clicking on the target, we are getting into the second view, which is bond disconnection view. By hovering over these bonds, you can see the names of the reaction that Synthia proposed to construct this specific bond. And by using this switch toggle here, you can switch the building blocks view to the pathway view. So actually we as chemists are used to this view, right? And here in this view, you can actually see the details of each of these reactions, the names, the conditions. This is a very interactive view. So we can click on the reaction name as well to get some more information, like for example, typical conditions or illustrative references for this type of chemistry in the literature, or also review the literature reactions that are similar to the one that was predicted by Synthia. And you can just like review all of this pathway just by exploring and clicking all of these items. They are very clickable. You can click on the book icon, for example, to get directly to this similar literature reactions that are in the database. And you can see as well like other details like prices of the molecules. You can also see click on the molecules to get some more information like PubChem link, like vendor information. So this is very useful. And by default, Synthia displays 50 pathways from millions of potential pathways, and the user can scroll through these pathways and also can filter the pathways out by limiting to specific reaction or intermediate he or she would like to use, or specific reaction type. And there are also additional possibilities that Synthia offers, for example, downloading the pathway in the PDF or RDF, or sharing analysis with other Synthia users, or for example, commenting on the analysis. And now I would like to talk about how Synthia can help you with the multi-target oriented synthesis. And there are two features of Synthia that are very important here. The first one is called Shared Path Library, and the second one is called Diversity Library. So let's move to the first library feature. This Synthia functionality enables to simultaneously plan a synthetic pathway for the entire library of target molecules in the multi-target oriented synthesis. And what Synthia does here, it prioritizes the routes with shared synthetic steps. As a Synthia user, you can upload into Synthia a library of target molecules you want to synthesize, and what Synthia will do is identify the synthetic pathways that have the highest number of common intermediates and starting materials, prioritizing the pathways with late-stage functionalization. This enables you to synthesize your molecule library in the most efficient way, utilizing as much as possible the common intermediates. So let's take a look at the real-life example of such a library. This is a library of a novel aminobutanoic acid-based inhibitors for the treatment of the lung cancer that was published in Journal of Medicinal Chemistry just like a few months ago. And what we did here, we entered this molecule into Synthia for it to design a shared pathway. And after just like a minute, the software came up with the pathway that included common synthetic steps for this entire library of 8 molecules. And this is how the results are displayed in the software, just like zoomed in one of the pathways. The common reactions and molecules are highlighted with a color, so you can quickly see how many steps in the pathways these pathways have in common for this entire target and library. And this is just another example, 12 molecules target library, and we did the same thing here. We uploaded this library to Synthia and started a common path search. So here you can see one more time my Synthia account with all of this analysis I've done. Let's click on the shared path library analysis for these 12 molecules from the slide. And here at first you can see this colorful schemes for the pathway. After clicking on the eye icon at the top, you can see all of the structures for this target library. And what you can also see, like for each of these targets, target 1, target 2, you see the schematic pathway view, right? So here's target number 4, number 5, and up until 12 targets from this library we entered. And this is as well like very interactive view. You can click on the reactions, you can click on the molecules to see them bigger, see some more details here. And let's take a look together at one of these pathways that Synthia proposed. Let's make it bigger. You can use these arrows here to enlarge the image, and you can see these colorful parts in these branches, in this convergent synthesis, right? So let's zoom in a little to this one branch, and here you can see the small number 12 next to the molecules and as well next to the reactions. So, and this is like that up until this intermediate, brown intermediate at the end. And let's take a look at the bottom branch. There is this yellow intermediate with small number 12 and up until the starting materials. And what it does and what it means is that these 2 branches were repeated for all of these molecules in their library. So we can make this intermediate and this intermediate in a bigger quantity and then reuse this for the synthesis of this entire library. Of course, you can change the visualization. You can, for example, leave only the highlight for the reactions, which makes it a little more clear. And this you can do for like all of this pathway that Synthia shows. So this functionality enables to quickly predict what is the most optimal synthetic strategy for synthesizing entire library of these target molecules with shared synthetic steps by prioritizing in Synthia the late stage functionalizations. Okay, so for human, it is sometimes very hard to come up with the pathway that is a common one, especially when the target molecule have functional groups that are compatible within different reaction conditions. But for Synthia, on the other hand, our algorithms can really screen billions of reactions within a very short time, coming up with the most optimal solution for this entire library. And by that, a chemist can actually synthesize one intermediate in a bigger quantity to quicker get all of these molecules in the lab, so it can really save a lot of time. Let's now move to the second feature I would like to talk about that will be released very, very soon, which is called Diversity Library. And this Synthia functionality will enable to automatically design target library based on the synthetic pathway. The user selects starting materials which he would like to diversify, and with this selection, Synthia generates a library of targets similar to the original one. What is extremely important to say here is that Synthia generates this library considering its entire power of reaction rules database, this huge database we have, with all of this context information of incompatible functional groups in every reaction, and also the power of algorithms that are removing non-selective reactions, non-existent motifs, and all of these other algorithms we have built inside our Synthia tool. So all of this together means that the library targets that are generated are chemically correct and synthesizable. So let's take a look one more time at my Synthia dashboard. So here I have all of this analysis. Let's click together at one of this automatic retrosynthesis analysis I did for lenacapavir. This is AIDS drug, pretty recent drug. Here you can see this building blocks view that I have here, right? And there's this small flask icon which says create diversity library. And here we are moving to this view where you can select the building blocks in which you would like to use for diversification. You can select all of them. You can select only a couple of them. You can select, for example, only the ones that are closest to the target for the late stage diversification. And then you can also select how many molecules you would like this library to generate. I selected 96 here and click Create Library. This search can take from a couple of minutes to a couple of hours, so I have this already pre-calculated. And let's maybe zoom in a little as well to have a better view on the molecule structures. And as you can see here, these molecules that were generated are having this like small yellow parts, and this is showing actually these points of the diversification between the original targets and these molecules that were generated in this target libraries, right? You see everywhere there is this like yellow part. And what Synthia offers here as well, you can click on the pathway view to explore the pathway in more details, to take a look closer at these building blocks that were diversified here, and also click on the reaction names. This is this interactive view. And Synthia here generated 96 targets, so the amount I requested the software to generate. And what you can also do, you can as well sort these targets by different criteria, like for example, similarity to original target or molecular weight. And what is very interesting here as well, that you can change the coloring options from the structural differences that I had selected at the beginning to the building blocks coloring. So this enables you also to quickly spot different building blocks that were used. Like for example, in the molecule number 12, you see this difluoromethane group, and in 13, you have trifluoromethane. So all of these differences, structural differences that were done to generate this diversified target structures. So there's also a question on how you can export these results in Synthia, and these results can be downloaded in many ways. You can export each of the target pathway in PDF or RDF for each of the molecules, but you can also do what I'm doing here right now. I'm selecting the molecules that I would like to choose. I selected 8 molecules, and these molecules you can then download either, for example, as an image or as the CSV file. And this CSV file have inside this target number from Synthia, but also the SMILE structure, so you can easily like copy it or upload to some other molecule discovery tool for further processing. And let's quickly move to Synthia APIs. There are 2 Synthia APIs versions that we have, Synthia Synthetic Accessibility Core API and the full retrosynthesis API. So let's start with the first one. This first Synthia API predicts how easy or hard it is to make a molecule in the lab based on the molecular structure. And what it does, it returns a score that approximates how many steps it takes to synthesize this molecule using commercially available building blocks. And what is very important to mention here as well, this scoring is very fast. It takes only 25 milliseconds per molecule. And the second type of API is this full retrosynthesis API, and these are actually Synthia results, Synthia's pathways, but in the form that can be connected with the internal molecule discovery tools for further processing or visualization if you want to do that. And there's also the shared path library available in this API, but with higher limits for number of molecules, and that you can upload there, which makes this extremely powerful. I know that Dr. Yu Yang will talk more about this in a moment, so I will stop here. And as the summary, what are these 4 main gains that one can get by using Synthia? The first one is reduction of the risk of overlooking a valuable pathway that would, for example, make your pathway shorter by 1 step, or using cheaper or safer starting materials. The second thing is reduction of time and cost of the synthesis during both design the initial pathway and also during development of production synthesis and optimization. The process chemists really like using Synthia to have this possibility to explore all of the synthetic options, customization options that Synthia suggests. The time savings as well are connected with sparkling innovation. So by providing innovative ideas for the pathways, Synthia sparks innovations in teams that are aimed to design this optimal solution, meeting the project needs. So think about it like as a calculator that is designed to streamline your work and help you to get the best idea faster. And this is what we also very often hear from many of our Synthia users, that Synthia accelerates this brainstorming in the teams. And the last thing, you can predict synthesizability to filter millions of molecules within a very short time, or connect Synthia results with the internal software via our Synthia full retrosynthesis API. What I would really like to mention here as well, there's a new product that was launched today actually, which is called MPredictor. And this is an AI prediction tool for crystallization. And this is a really amazing tool. So please feel free to check our website here, which you can scan here through this code, and get in touch with us via the contact form. We are very happy in the Synthia team to talk to you about how Synthia can support you, your synthetic workflows, how we can help you to simplify the pathway design or molecule design and enable you to get your target molecules faster, as quickly as possible and as efficiently as possible. So please feel to contact us. There is this webpage, synthiaonline.com. So you can visit this website. There's this contact form that you can use. And we are very happy and looking forward to talking to you about how we can help you. With that, thank you so much. And I will pass to Dr. Yu Young.
And very quickly before we go to you, Young, we're actually going to have another poll, but thank you to Eva for that really comprehensive overview of the software. If you have questions for Eva, of course, she's here live, so she will be able to answer those towards the end of the webinar. I can see people are already finding the questions box and sending things in, so please keep the questions coming. I'm going to bring up a quick poll. Well, there we go. Just bring that up for you now. You can click on the screen to interact with the poll. And the question is, which outcome would be the most valuable to you using a retrosynthesis software? You may need to use the little scroll bar to see all of the answer options there to click on. Again, I don't know how it looks for you. As an audience member, it will depend on which platform you've joined us on, if you're on a phone or a computer or a laptop, whatever it is. But thank you. I can see lots of you have already found the poll there, putting in your responses, which is brilliant. It seems at the moment that planning synthetic routes faster is the most popular response, which is probably not a surprise, but we've got quite a lot of people designing molecules using AI and also generating new ideas, which is great. That's great to see. Thank you all for joining in with the poll. It's nice for our speakers to know, again, who's in the audience and what they're trying to do with this software, what they want to do, what they are using it for. And great, I can see some more questions coming in, so thank you. And we're going to now move on to Yu Young, having heard from Eva already. Again, this is a pre-recorded video, but as I've said, Yu Young is here with us live, as is Eva, so please keep those questions coming in. But we'll hear Yu Young's pre-recorded presentation now, and I'll be back in a moment.
Thank you, Natalie, for the introduction, and thank you, Eva, for the overview of Synthia. I am Yu Young Song, a principal AI scientist at Standigm. Today, I will discuss how we integrate Synthia into our drug discovery workflow from an AI scientist perspective, highlighting our collaborative efforts between the AI Research Center and Drug Discovery Center to effectively assess molecule synthesizability. Standigm's mission is to transform drug discovery through cutting-edge AI, accelerating the journey from target identification to market-ready therapeutics and driving innovation in healthcare. The name Standigm reflects our goal, combining standard and paradigm to set a new standard in the AI-driven drug discovery landscape. For this mission, our multidisciplinary leadership fosters collaboration across AI, biology, chemistry, and drug development. Close collaboration between the Drug Discovery Center and AI Research Center allows Standigm to integrate computational and experimental expertise to accelerate the development of novel therapies. Here, I show a brief history of Standigm. Our journey began in 2015, aiming to innovate AI in drug discovery. Key milestones include launching Standigm Expander in 2017 for drug proposing, Standigm Best in 2018 for novel compound design, and completing our full-stack AI platform for Standigm Ask in 2020 for target discovery. Looking ahead, our focus for 2024 is on commercializing our products and expanding into cloud-based SaaS, making our technologies globally accessible. Now, I will walk you through Standigm's AI-empowered drug discovery process and outline how the Drug Discovery Center and AI Research Center collaborate. We will explore how Synthia is integrated at various stages to support both centers, enhancing the efficiency of our workflow. At Standigm, we enhance the traditional drug discovery process through close collaboration between Drug Discovery Center and AI Research Center. Standigm utilizes two major AI platforms, which is Ask, a biology AI platform that aids in novel target identification, and BEST, a chemistry AI platform designed for novel compound design. In the early stages, our Drug Discovery Center with experts in biology and chemistry selects disease targets based on their knowledge and experience supported by the Ask platform. Once the targets and hits are selected, the AI Research Center uses Stella within BEST, a multi-objective lead optimization platform, to design and refine drug candidates. Stella leverages various tools including QIP-ADME for ADMET prediction using quantum chemistry insights, GT-AutoML for automatic SAR model generation, and SpeakUp for assessing patent novelty. For assessing synthesizability, we use our own custom synth filter to remove molecules that are designed to be unsynthesizable, and Synthia to ensure that the remaining molecules can be feasibly synthesized. After the Drug Discovery Center experiments with the designed candidates, feedback is provided to the AI Research Center, which uses this feedback to refine the next generation of molecules. This human-in-loop, expert-in-loop approach ensures continuous improvement through close collaboration between both centers. Now, let's take a closer look at how Standigm assesses synthesizability with Synthia working alongside Stella. First, the Drug Discovery Center identifies targets and key molecules and passes them on to the AI Research Center. At this point, the AI Research Center uses Stella to design the drug candidate molecules based on these targets. Once the designs are optimized, they are sent back to the Drug Discovery Center. Stella is a powerful lead optimization platform driven by genetic algorithms and AI. During the optimization process, molecules go through multiple cycles of generations, filtering, and selection. This ensures that the right molecules are chosen for multi-parameter optimization, aligning with the project's goals. In this process, the Synthia SAS API, along with our internal synth filter, plays a crucial role in filtering out molecules that are difficult or impossible to synthesize. After the generation, Synthia Full Retro API is applied to further screen the selected molecules for synthesizability before they are delivered to the Drug Discovery Center. Once the optimized molecules pass through screening, they are reviewed by computational chemists and medicinal chemists in Drug Discovery Center. Using their expertise, database, and detailed synthesis pathways from Synthia web app, they assess which molecules are most feasible for synthesis. Synthesis results are then shared with the AI Research Center, allowing for improved generations in the next cycle. In this section, we will explore case studies that demonstrate how Synthia has been integrated into Standigm drug discovery workflow. I will highlight its impact on filtering and screening processes, as well as the improvements in synthesizability observed before and after its implementation. This diagram illustrates how the candidate molecules designed by Stella and in the AI Research Center are evaluated and delivered to the Drug Discovery Center for synthesis. We apply Synthia at multiple stages in our drug discovery workflow, particularly for evaluating synthesizability. Synthia's tools, including the SAS API, Full Retro API, and web app, play critical roles in our rigorous filtering and screening processes. First, the AI Research Center leverages Synthia by Stella to generate over 1 million molecules. To narrow down this vast number, we apply filters to identify and eliminate molecules that are impossible to synthesize. We also use Synthia SAS API, which assigns a synthetic accessibility score to filter out molecules that are likely to be unsynthesizable. This allows us to focus on the more feasible compounds. For the remaining molecules in each cycle, we calculate key properties such as ADMET to evaluate their potential as drug candidates. During this MPO phase, we prioritize molecules that not only demonstrate strong biological activity, but also have a high likelihood of successful synthesis in the lab. Through this filtering process, we reduced the pool to approximately 1,000 to 2,000 molecules. The Synthia SAS API assigns a score from 0 to 10 for synthetic accessibility. By inputting a SMILES list, we can obtain the score list within minutes, allowing for rapid decision-making. After deciding to integrate Synthia SAS API into Standigm's drug discovery workflow, we conducted several experiments to determine how best to leverage the Synthia Synthetic Accessibility Score. Today, we are sharing the results of a study with about 870 Standigm molecules that are reviewed by Standigm's medicinal chemists for their synthesizability as of late 2022. These molecules are derived from 3 of Standigm's pipeline candidates, each with a different scaffold. We compared the RDKit assay score and Synthia SAS results. The blue dots represent molecules that are successfully synthesized, while the light gray dots indicate molecules that were either deprioritized due to time and cost considerations or failed during the initial synthesis attempt. As you can see in these 3 graphs, molecules with lower assay score and Synthia SAS scores were more likely to succeed in synthesis. Neither score is always accurate. However, when both scores are high, we decided to deprioritize those molecules for synthesis, focusing on more promising candidates. In conclusion, using the Synthia SAS API allows us to efficiently filter out molecules that are not promising and difficult to synthesize during the candidate design via Stella, thereby streamlining the drug discovery process. Here, we are at the final step before delivering the Stella design molecules to the Drug Discovery Center, the screening stage. At this stage, we select around 200 candidates from the 1,000 to 2,000 generated molecules that best align with the project goals and demonstrate high potential for both synthesizability and novelty. To assess synthesizability with greater accuracy, we utilize the Synthia Full Retro API for a thorough evaluation. Synthia Full Retro API generates retrosynthesis pathways similar to the Synthia web app. The process is straightforward. Input a SMILES list and pathway calculation parameters and receive retrosynthesis pathway information as text, which includes the pathway score. To the best of my knowledge, the path score takes into account factors such as the number of synthetic steps, whether the starting material are known or available for purchase, their cost, and other related factors. We hypothesized that this scoring system will correlate well with the synthetic feasibility and prioritization process used by Standigm's medicinal chemists. To test this, we evaluated the same 870 Standigm molecules as in the previous studies. Synthia Full Retro API or web app suggests up to 50 pathways for each molecule, and we use the lowest path score for each molecule to create a histogram of their distribution. As shown in the histogram, molecules that are successfully synthesized and prioritized by medicinal chemists have lower path scores represented in blue. This indicates that Synthia's path score can be a valuable metric in screening molecules for synthesizability. Since each retrosynthesis calculation takes a few minutes per molecule, it is not feasible to use the full retro API for large-scale generation processes. Therefore, it is ideal for screening before delivering the selected molecules to the Drug Discovery Center. This collaborative workflow helps the AI Research Center select more promising molecules and ensures that the Drug Discovery Center receives candidates with a high likelihood of successful synthesis. Now, after the screened molecules reach the Drug Discovery Center, about 200 candidates undergo an expert review. During this step, further assessments, such as docking pose analysis, are performed by computational chemists, and medicinal chemists review the synthetic feasibility of the molecules based on their knowledge, experience, and the retrosynthesis paths calculated by the Synthia web app. Once these molecules pass the review stage, a few dozen selected candidates are evaluated for their synthetic complexity using Standigm's internal synthesis rank system. Based on this ranking, the team proceeds with the synthesis of the most promising candidates in a step-by-step manner. The Synthia web app offers medicinal chemists more flexibility compared to the full Retro API by allowing them to adjust various options in the retrosynthesis calculations. For example, they can evaluate criteria for starting materials and identify key intermediates shared across similar structures. Before we initiate synthesis, we need to decide which molecules will be prioritized for synthesis attempts. For this purpose, we evaluate the synthesis rank. The Standigm synthesis rank used by medicinal chemists is designed to improve the time and resource efficiency of synthesis. This rank evaluates multiple factors that affect synthesis difficulty, including the number of synthetic steps, the number of chiral centers, the availability of building blocks, and known chemistry references or reactions. The molecules are categorized into 5 levels, ranging from rank 0, which is impossible to synthesize, to rank 4, which indicates molecules that are readily synthesizable. When we plotted the synthesis rank histogram for about 1,000 Standigm molecules evaluated by the medicinal chemist as of 2024, we saw that the majority of molecules that are ultimately synthesized fall under ranks 2, 3, and 4. This shows that we tend to synthesize molecules with these levels of synthetic difficulty. The evaluation of synthesis rank is time-consuming because medicinal chemists rely on extensive data and experience. It's not just about determining whether a molecule is synthesizable or not, but it is about judging which molecules are more likely to succeed in synthesis. After adopting Synthia's, Standigm AI Research Center and Drug Discovery Center aim to optimize the synthesis process even further. By leveraging Synthia, the team sought to proactively collaborate on determining synthesis feasibility. Although each project has different criteria, the main goal was to focus on delivering rank 2, rank 3, and rank 4 molecules with higher synthesis potential to the Drug Discovery Center for synthesis attempts. To accomplish this, the AI Research Center sought to approximate Standigm synthesis rank using Synthia-generated data such as path scores and the number of steps. During both at 2022 evaluation and in 2024, we found that using Synthia path score helped us effectively pre-screen molecules that were likely to be ranked rank 0 or rank 1. These molecules had low synthesis potential and were less likely to proceed to actual synthesis in the Drug Discovery Center. We can observe how the path score distribution varies for 1,000 Standigm molecules as of 2024, calculated using Synthia Full Retro API. The distribution varies by synthesis rank, allowing us to screen out molecules with lower synthesis potential based on their path scores. So the molecules which are represented in red and orange here. Since each project involves different scaffolds, there is no fixed cutoff score for all cases. Instead, we adjust the cutoff dynamically based on early feedback from the Drug Discovery Center for each project. After completing the review and synthesis rank evaluation, the final step is actual synthesis, followed by continuous refinement and iteration of the process based on the synthesis outcomes. Thus, by utilizing Synthia's API and web app at different stages, SAS for rapid filtering, Full Retro API for more detailed screening, and web app for medicinal chemists, we have been able to clearly define the roles for each tool. This has improved collaboration between centers, enabling us to efficiently deliver candidates with higher synthesis potential. After evaluating Synthia, we integrated into our drug discovery workflow over the past 1.5 years, and here are the results. The AI Research Center generates molecules using Stella, and after it is delivered to Drug Discovery Center, In the expert review stage, computational and medicinal chemists evaluate these molecules. By utilizing Synthia's information, the number of molecules each medicinal chemist could review has increased by approximately 40% with the same effort and time, leading to more efficient reviews. Furthermore, in terms of evaluating the synthesis rank of molecules before attempting synthesis, we observed a 16% reduction in the proportion of molecules rated as rank 0 or rank 1 after incorporating Synthia. This means that utilizing Synthia at various stages, the AI Research Center has been able to deliver molecules with higher synthesis potential, providing tangible support for medicinal chemists. As we move forward, Standigm aims to explore synthesizable and novel compound spaces more effectively. Currently, Stella conducts multi-parameter optimization based on ADMT, STAR, and other factors to design and explore the optimal compound space that aligns with project goals. However, synthetic feasibility and patentability are carefully assessed during the screening phase after generation is completed. By leveraging the combined strengths of Synthia and Spica for synthetic accessibility and novelty within our generative model, we can envision expanding our capacity to discover the high-quality drug candidates. Now let's summarize how Synthia has been integrated into Standigm's drug discovery workflow successively. Synthia provides comprehensive support across multiple stages of our workflow. From filtering molecules through the SAS API, detailed screening using the full Retro API, and final review with the web app, we have successfully streamlined the evaluation of synthesizability. Synthia integration has led to measurable improvements. So we have seen an enhanced ratio of synthesizable molecules and a reduction in unsynthesizable molecules delivered for synthesis. Collaboration between AI Research Center and the Drug Discovery Center has been instrumental, improving decision-making and advancing our projects more efficiently. Looking ahead, we aim to leverage Synthia alongside Spica to further explore and discover synthesizable and patentable compound spaces. I really want to emphasize that integrating Synthia has brought tangible benefits in terms of efficiency, collaboration, and overall project success at Standigm. Thank you for joining me today. Thank you very much.
That was brilliant. So thank you to Yi-Young. That was just, great to see the case study and all that detail. I'll just remind the audience, if you need to watch again, if you missed anything or you just want to go back and take in all that information, then we will be sending a recording of the webinar out to everyone here within the next few days. So please look out for that in your inboxes. You can definitely go back and watch again if it's there's been a lot of information. So that's great. I'm going to bring up our final poll right now. So I'll launch that right now. Again, you can scroll to see all the options for your answers. How would you use Synthia in AI-driven molecule or drug discovery? So you've got lots of different options there. If none of those apply, you don't need to vote, obviously. And if you want to let us know more, then do pop a little message into the questions, into the chat box, and we'll see that as well. So we'll just give you all a few moments to have a look, and then we're going to move on to the questions with our speakers. So thank you to everyone that's sending in questions. There's a lot coming into the questions box, which is lovely to see. We might not get time to get through all of the questions, but I think my team have shared in the background on the chat box how you can contact the companies. So please use those. If you've got really specific questions, that's often a better way to do that, and you'll get some good responses from them. But we'll see what we can get through today. I can see my colleague has just shared that link again. Right, okay, so I will quickly end and share the results. I'll give you a couple more moments. There we go. So it looks like evaluating AI-generated molecules with Synthia is the most popular response there, which is good to see. So I'm going to close that poll now. Thank you for all voting on that, if you did. So I'm going to welcome Eva and Yi-Young. Right, welcome back. Right, let's see if we can get through some of these questions. We've got lots coming in. A good one to start with is perhaps how customizable are the synthesis routes generated by the software? And whoever wants to take that question, and you can both have a little go at answering that if that works for you.
Yeah, I can take that. So Synthia has a lot of customization options that you can use, and they're both for example, excluding hazardous reactions or hazardous reagents, for supporting sustainability, but also to take into account your needs in your synthetic projects. Like, for example, what kind of starting materials you would like to use. If you would like to go through generate the pathway that goes through a specific intermediate, for example, or if you are considering the quantity of starting materials, their availability, right? So to start only from the molecules that are available in a very high quantity. So there are like a lot of options that you can use. Also, you can, for example, ask Synthia to remain intact specific functional group or structural motif in your target molecule, so that it means that it will take this specific piece just like from the starting material. So there are very like many advanced options that you can fine tune in the software to make your pathways according to your needs, to your synthetic needs.
That is great to know. Thank you, Eva. Okay, we've got a question for you, Young. Do you know what made Standigm pick Synthia as a platform, and how did Merck work with you to get you up and running?
So the question is why we picked Synthia and how we worked with Merck. So at the beginning, when I joined Standigm, we were trying to find out a platform which will support medicinal chemists and help to evaluate synthesizability. We went through several platforms, and Synthia was the one that was picked by our medicinal chemist. So for AI scientists, we wanted some kind of API which is fast working, but which was more important is this acceptable to our medicinal chemists? Because if we do the API is fast enough, if the medicinal chemist doesn't understand and don't like this result, it was not suitable for us. So at that time, we asked Merck Synthia team that if they can make API for us. So they didn't have API service at the time, and they did it. So we worked with them for about a year, and they made a beautiful full lateral API, and we are now using those full lateral API in our drug discovery workflow.
Oh, that's amazing. Brilliant. Thank you. Thank you for that insight. That's really good to hear. And how these two companies working together is just lovely. Okay, a question. How can Synthia assist in optimizing reaction conditions for complex molecules? Can it do that? And if so, how?
So that's a very good question, right? So Synthia is rather a tool that you can consider as generating ideas of how you can make your molecule, a specific step, right? If you want to perform specific reaction, right, from your substrate to the product, Synthia can help you to see various methods, right, on how you can actually do this step, right? So you can see various publications that are referencing this specific reaction type. So it can help you basically to see what kind of reaction conditions were used, what kind of reaction types were used to perform a particular step.
Okay, thank you. That's great. We're getting through the questions here. Lots more coming in still, which is lovely. I think this one's probably for Eva, but let's give this one a go. Can Synthia design pathways to molecules that have never been published, like, for example, novel unpublished drug candidates?
Yeah, that's a very good question. And this is actually, to be honest, the most important way of how the software is being used, right? Because due to our reaction rules database and all of these algorithms we have, Synthia is not biased on the literature, on only the molecules that were synthesized before, but can create chemically correct synthetic pathways to the molecules that have never been synthesized, actually due to this power of the reaction rules that were generated based on the reaction mechanism, not on like a specific molecules that only were published within the specific methods. So basically this makes the software very useful to novel molecules never synthesized before, that even sometimes have not a similar synthesis in the lab.
I see. Okay, that's good to know. Thank you. Okay, a question for you, Young. I'm trying to split them as evenly as I can, and we've got lots coming in still. How do you integrate Synthia's retrosynthesis predictions into your drug discovery workflow, Stella? Could you provide a detailed example of how you use the Synthia API and web app in your process?
Okay, thank you for a great question. So in our AI-driven drug discovery workflow, we integrate Synthia's retrosynthesis predictions with our generative model, which is Stella. And through an iterated feedback loop with our medicinal chemists and AI scientists, we try to enhance that. So for example, Stella focusing on improving overall molecular properties. They may generate many similar structures by attaching substructures or functional groups to a fixed scaffold. And while these modification enhance properties, they sometimes result in structures that are difficult to synthesize or lead to undesirable synthetic routes by our medicinal chemist determination. So in this case, our medicinal chemist identifies such issues, and with the help of Synthia's predicted routes, as Synthia provides very numerous adjustable calculation parameters, chemists select and let AI Research Center know which substructures or structures or functional groups are undesirable. And then with this kind of advice, we instruct Stella to skip molecules with these undesired substructures or structures in future generations. And then we can analyze alternative pathways and their associated Synthia scores when we do screening in the next generation. So it's a kind of iterative process.
Okay, that's lovely, thank you. Okay, quick question about IP. So who owns the IP generated from the novel molecules, and does Merck have a stake in that?
Amazing question, and I heard this question like a lot over the last years, and I can say that like entire intellectual property is owned by the customer that uses Synthia. So we as Merck don't have any IP, anything in terms of the IP ownership for the molecules or the pathways that are generated using the tool.
Okay, that's great. Thank you. That's a very solid answer. That's good to know. Okay, I think we are rapidly running out of time. I'm going to end with this question because it's quite a nice question about collaboration within teams. Is there a way to share the results with colleagues within the platform if you're working on a similar target? And I think that's probably a good question for Eva, but you, Young, you might have had experience if that's possible. So.
Yeah, so we have collaboration tools within the Synthia solution. So basically, you can share a synthetic pathway within the different users of Synthia from your team, but you can also comment on the results. There is a very nice commenting box within the Synthia tool that allows you to, write notes about your pathway or write what you like about it and just like store this for the future. And also this information then is being shared with the person that you shared this analysis with. So it can really enhance the collaboration if you're working on the same molecule and you want to get the best pathway as quickly as possible. You want to exchange notes on this pathway. This particular feature allows you to do that. So that there are very good collaboration tools within the software.
That's brilliant. You, Young, you were nodding a lot there, agreeing with what Eva was saying. Do you want to share just a little bit of your experience with that? Has that been really useful for your team?
So yes, this sharing interface was developed recently, and this was one of the things that we have suggested Synthia team to make up. And we are having meetings regularly, and they listen to us and upgrade the interface every time. So I think that's the point, where the Synthia has their strength.
Oh, that's so good. It's so good to hear that they're taking that user feedback and improving things. That's really good to see. I'm glad that that's working for all of you. Well, thank you so much. I'm afraid we're literally out of time. So that's all we have time for today. Sorry, I'm really sorry if we didn't get to your question, but you do have the contact form in the chat box there. So please do contact, Merck or the speakers if you need to, and they will be able to get back to you and help you out with anything that you're looking for. But thank you so much to Eva and you, Young, everyone for joining us to share so much valuable information. It was great to hear from you both. And we'd like to thank Merck again for sponsoring this webinar. We really value them choosing to work with Chemistry World again. If you missed anything today, again, if you want to go back and watch again, you're going to get a recording in the next few days, so please sit tight until that arrives in your inbox. And finally, thank you to you, our audience. You've got really involved in the conversation today, answered our polls, and that was great to see. So thank you very much for getting involved. But from everyone here at Chemistry World, we look forward to seeing you again in a future Chemistry World webinar. Thank you very much.
Thank you so much.
Thank you so much.